Garden plants seem to vanish when winter arrives, but they are far from dead. Many gardeners assume their perennials have simply stopped growing, yet beneath the surface and inside every cell, an intricate survival mechanism is unfolding. Plants cannot migrate or seek shelter, so they have evolved strategies that would astound even experienced growers. What appears to be dormancy is actually a carefully orchestrated biological transformation that begins weeks before the first frost and continues throughout the coldest months. Understanding this process changes how gardens are managed in autumn, what protection is truly needed, and why some plants thrive after harsh winters while others struggle. This is not about guessing when to mulch or hoping for the best. It is about recognizing the science behind winter survival and working with it. Dormancy is not sleep in any human sense. It is an active state where plants intentionally slow their metabolism, redirect resources, and fortify cells against freezing temperatures. This process, called senescence, starts when day length shortens and temperatures drop consistently below 50 degrees. Chlorophyll production halts, revealing the yellows and reds that were always present but masked by green pigments. At the same time, plants begin moving sugars, starches, and proteins from leaves and stems down into roots and woody tissue. This is not decay, it is strategic resource allocation. Hormones like abscisic acid increase, triggering the formation of a corky layer at the base of each leaf stem, which seals off the connection and allows the leaf to drop without leaving an open wound. The timing is critical. If this happens too early, the plant loses valuable photosynthesis time. Too late, and an unexpected freeze can damage unprepared tissue. Most temperate zone plants have evolved to read multiple environmental signals, temperature, day length, and even soil moisture, to time this transition perfectly for their native climate. Deciduous trees and shrubs demonstrate the most visible winter preparation. As autumn progresses, sap movement slows dramatically, and water content in branches drops by as much as 50%. This dehydration is deliberate. Water expands when it freezes, and cells full of liquid would rupture under ice crystal formation. By reducing internal moisture and concentrating sugars and other solutes in remaining fluids, plants lower the freezing point of their sap, much like antifreeze in a car radiator. Some species, like birch and willow, can survive temperatures as low as 40 degrees by accumulating sugars to levels that would be toxic during the growing season. The bark thickens slightly as cells harden, providing insulation and a physical barrier against desiccating winter winds. Meanwhile, buds that will produce next spring's leaves and flowers are already formed by late summer, wrapped in protective scales coated with resinous substances that repel moisture and prevent premature opening during brief warm spells. These buds remain in a state of suspended animation, requiring a specific number of chilling hours, typically between 32 degrees and 45 degrees Celsius, before they can break dormancy in spring, which prevents them from responding to every temporary thaw. Evergreens face a unique challenge. They keep their leaves throughout winter and must continue minimal photosynthesis while protecting tissue from freezing and desiccation. Needles and broadleaf evergreen foliage are coated with a thick, waxy cuticle that reduces water loss. And stomata, the pores that allow gas exchange, close tightly when temperatures drop. Many evergreens also change needle color slightly, taking on bronze or purple tones as anthocyanin pigments accumulate, which act as a sunscreen to prevent photooxidative damage when bright winter sun hits frozen leaves that cannot process light efficiently. Root systems of evergreens continue slow water uptake even when soil temperatures hover just above freezing, but the rate is so minimal that winter burn can occur if dry winds or intense sun cause more water loss than roots can replace. This is why evergreens often show browning on the windward side or sun-facing portions in late winter, especially in exposed locations. Rhododendrons and other broadleaf evergreens curl their leaves tightly when temperatures drop below 25 degrees, reducing surface area exposed to air and creating small pockets of still air around the leaf that act as insulation. This curling is reversible and can happen multiple times throughout winter as temperatures fluctuate. Root systems do not go fully dormant as quickly as above-ground growth. As long as soil temperatures remain above 40 degrees, roots continue to grow slowly, absorbing nutrients and water and even producing new fine root hairs. This late-season root growth is why autumn is often considered the best time for transplanting trees and shrubs. They establish below ground while top growth is paused. Once soil temperatures drop below 40 degrees, root activity slows dramatically but does not stop entirely. Even under snow, soil temperatures rarely match air temperatures. An insulating layer of snow or mulch can keep soil 10 to 20 degrees warmer than ambient air. Roots rely on stored carbohydrates produced during the previous growing season, and plants that were stressed, poorly fed, or damaged by drought in summer often struggle in winter because they lack adequate reserves. Root hardiness varies significantly by species. Some perennials can tolerate root zone temperatures down to 0 degrees, while others, like lavender or certain salvias, begin suffering root damage at 20 degrees. This is why zone ratings matter. They reflect the coldest temperature roots can survive, not just what top growth can tolerate. Herbaceous perennials take winter preparation to an extreme by dying back completely above ground and storing everything in roots, rhizomes, tubers, or bulbs. Hostas, daylilies, peonies, and ornamental grasses pull nutrients from leaves back into roots during autumn, then allow foliage to collapse when hard frost arrives. This above-ground death is protective. There is no living tissue exposed to freezing winds, desiccation, or foraging animals. Underground storage organs are packed with starches and sugars that serve both as energy reserves and as cryoprotectants. True bulbs, like tulips and daffodils, are essentially compressed stems surrounded by fleshy scale leaves, and they require a cold period of 12 to 16 weeks below 45 degrees to trigger flowering mechanisms for spring. This is why bulbs planted too late or in climates that do not provide sufficient chilling often produce foliage, but no flowers. Tubers like dahlias and tuberous begonias are less cold hardy. Their cells lack the same antifreeze compounds, and they must be dug and stored above freezing or heavily mulched in marginal zones. Some perennials, like hellebores and winter-blooming heathers, reverse the pattern entirely and remain semi-active through winter, even blooming during thaws, because they evolved in climates with mild, wet winters and hot, dry summers. They treat winter as their growing season. Cold hardening is a gradual physiological change that occurs over weeks, not days. As night temperatures begin dipping into the 40s and 30s in autumn, plants start synthesizing protective proteins, adjusting cell membrane composition to remain flexible at low temperatures and accumulating compatible solutes like proline and glycine betaine that stabilize proteins and prevent ice crystal formation inside cells. This process is triggered by both temperature and day length, and it happens in stages. The first light frosts in early autumn cause partial hardening. Plants can tolerate 25 to 28 degrees Celsius without damage. As cold exposure continues and temperatures drop further, full hardening occurs, allowing survival down to 0 degrees or lower, depending on species. However, this hardening can be reversed during warm spells, which is why late winter freezes are often more damaging than midwinter cold. A week of 60-degree weather in February can trigger dehardening, causing buds to swell and sap to flow, and if temperatures then plunge to 10 degrees, severe damage occurs. This is a major concern with fruit trees. A mild January followed by a February deep freeze can destroy an entire crop of flower buds that would have survived steady cold. One of the most common mistakes is cutting back perennials too early in autumn. When foliage is removed before plants have completed nutrient transfer to roots, they enter winter with depleted energy reserves. Waiting until foliage is fully yellow or brown, or even leaving it standing through winter, ensures maximum nutrient recovery and provides additional insulation. Another error is applying high nitrogen fertilizer late in the growing season. Nitrogen promotes soft, succulent growth that does not have time to harden off before frost and is highly susceptible to freeze damage. Fertilizer applications should stop by late summer with a shift to potassium-rich formulations that support cell wall strength and cold hardiness. Overwatering in autumn is also problematic. Saturated soil stays cold longer in spring, delays root activity, and increases the risk of root rot if drainage is poor. Conversely, plants entering winter drought-stressed are more vulnerable because dehydration is part of the hardening process, but it must be controlled. A deep watering before the ground freezes is ideal. It allows plants to hydrate tissues one last time before water becomes unavailable. Finally, wrapping or covering plants prematurely can trap warmth and delay hardening, leaving plants vulnerable when protection is removed or when temperatures drop suddenly inside the covering. Gardeners can support winter preparation by timing interventions correctly. Mulching should wait until the ground has experienced several hard freezes. Applying mulch too early keeps soil warm and delays dormancy, and it can provide cover for rodents that damage roots and bark. A 2 to 4 inch layer of shredded leaves, straw, or wood chips applied in late November or early December insulates soil, moderates temperature fluctuations, and reduces frost heaving, the cycle of freezing and thawing that can push shallow-rooted plants out of the ground. For marginally hardy plants, additional protection like burlap windbreaks or evergreen bough covers can reduce desiccation without creating overly warm microclimates. Watering evergreens thoroughly before freeze-up is critical, especially in dry autumns, because they continue losing moisture through foliage all winter. Pruning should generally be avoided in autumn. Wounds do not seal when growth is paused, and exposed cuts can dry out or become entry points for disease. Spring is the better time for most pruning after the worst cold has passed. For tender perennials in containers, moving them to unheated garages or sheds where temperatures stay between 35 degrees and 45 degrees provides enough cold for dormancy without risking root freeze, which is more likely in pots than in-ground plantings due to limited soil volume. When temperatures drop below a plant's hardiness threshold, ice formation inside cells becomes lethal. Ice crystals puncture cell membranes, and when tissues thaw, fluids leak out, causing that characteristic water-soaked blackened appearance of frost-damaged leaves and stems. However, many plants can tolerate extracellular ice, ice that forms in spaces between cells rather than inside them. As extracellular ice forms, it draws water out of cells through osmosis, concentrating solutes inside the cell and lowering the internal freezing point even further. This controlled dehydration is part of the survival strategy, and it is why slow freezes are less damaging than rapid temperature plunges. Slow cold allows time for water to migrate out of cells safely. In extreme cold, some woody plants can supercool their tissues, keeping water in a liquid state below its normal freezing point through the absence of nucleation sites, where ice crystals would begin forming. This is a precarious state. Any physical shock or internal nucleation event can trigger instant, catastrophic freezing. Snow cover is one of the best protections during extreme cold because it acts as an insulator. Even a few inches of snow can keep soil and low-growing plants 15 to 20 degrees warmer than exposed areas. This is why alpine and subarctic plants often survive temperatures far below what they could endure without snow. Spring awakening is not triggered by a single warm day. Plants track accumulated warmth, often measured in growing degree days, the cumulative number of degrees above a base temperature typically 50-degree force. Only after this threshold is reached do buds begin to swell and roots resume active growth. This internal accounting prevents premature growth during temporary thaws. Day length also plays a role. Many plants have photoreceptors that detect increasing light duration, which signals that true spring has arrived. Soil temperature is often more important than air temperature for perennials. Roots do not activate until soil warms to at least 45 degrees, which can be weeks after air temperatures are consistently in the 60s. This lag is why early spring can feel deceptive. Trees and shrubs may show green buds, but perennials remain dormant below ground. The sequence of emergence is predictable. Bulbs and early perennials like hellebores and crocuses respond first, followed by deciduous tree buds, and finally warm-season perennials like hostas and ornamental grasses. Watering needs change rapidly as growth resumes. Plants shift from minimal water requirements to active uptake within days, and neglecting this transition can stress newly emerging growth. This is also when winter damage becomes visible. Bark splits, dead branch tips, and browned evergreen foliage are only apparent once growth resumes, and the contrast with healthy tissue is obvious. Winter dormancy is not a passive state, but an active survival mechanism honed over millennia of evolution. Plants orchestrate cellular changes, reallocate resources, and respond to environmental cues with precision that rivals any engineered system. Gardeners who understand these processes can time autumn tasks, choose appropriately hardy plants, and recognize when intervention helps versus when it interferes. Observing how plants prepare for winter provides insight into their needs during the growing season. Stressed, poorly fed, or drought-damaged plants enter dormancy weakened and are less likely to survive or thrive the following year. Every autumn is preparation for the next spring, and every spring reveals the success of that preparation. For those looking to deepen their gardening knowledge, exploring plant physiology and cold hardiness science offers practical benefits that go far beyond memorizing zone numbers. If this perspective on winter survival was useful, consider exploring more about soil biology, seasonal nutrient cycles, or how to select plants that match not just hardiness zones, but also local winter patterns.